Hello, welcome to our first lecture for Exam 2 material, Microbial Genetics. So let's start off with this pretty cool image right here. This is an E. coli cell, so it's an E. coli bacterial cell. All of this yellow stringy stuff around it is its chromosome, or its DNA. Let me grab my pen. It's chromosome or its DNA. And there's a lot of it. And normally this would all be condensed inside the cell, just like our genomes are tightly compact and condensed within our cells. But when a bacterial cell experiences some type of negative environmental pressure, it can actually just barf out its genome like this, and some other bacterial cell can come along and eat that genome, essentially, and use parts of that genome that are useful for it. Pretty neat, right? But there's a lot of this, so if you just imagine that you took all this this yellow stringy stuff and you stretched it out as far as it would go, you would see that this genome is a giant circle. Because we're talking about a prokaryote here, we're talking about bacteria, they have circular genomes. And if you took this genome and you cut it, so it wasn't a circle anymore, and you stretched it along a ruler, you would see that their genomes are typically about 1 millimeter long. And you can see a millimeter on a ruler. So that's a lot of genetic material. Now, let's think eukaryotes and let's think humans. If you took all of the genetic material from a human cell, one human cell, and you stretched it out along a ruler, it wouldn't fit on a ruler. It would be over 2 meters long. That's a lot of genetic material. And that's just from one cell. We're multicellular. So if you took all of the genetic material from every single cell in your body and put it end to end to end to end to end, it would be over 67 billion miles long. That's enough for over 150,000 trips to the moon and back. So we have a lot of genetic material, and that should give you a pretty good idea of how complex we are compared to a prokaryote or a single-celled organism. That being said, as we go through and talk about DNA and RNA and DNA replication, we'll see a lot of these processes are actually highly conserved. So the way that we replicate our genome is really very similar to how a prokaryote replicates their genome as well. Before we can start talking about genome replication and genetic expression, we really have to understand these two forms of nucleic acid. We have RNA and DNA. Both of these are types of nucleic acid. DNA stands for deoxyribonucleic acid, DNA, and RNA stands for ribonucleic acid, RNA. And there's some important differences between these two nucleic acids. Both have these 4 bases. So DNA has adenine, guanine, cytosine, and thymine, and RNA has adenine, guanine, cytosine, and uracil. So where DNA has thymine, RNA has uracil. Also take note that DNA is double-stranded, so it has 2 strands, and it's in something called a double helix. That is the shape. Sorry for the bad handwriting. That's the shape of this structure. It's a double helix. RNA is single-stranded. So starting off with just that basic understanding, we'll go from there. Now imagine that the E. coli cell on the previous slide had not barfed out all of its genetic material. This is kind of what it would look like. So recall that in a prokaryote, or a bacterial cell, all of the genetic material is right inside the cytoplasm, and it is not encased by a membrane. There is no membrane around this genetic material. Nothing. So it's called a nucleoid, not a nucleus. So make sure you remember the distinction between those two. Prokaryotes have a nucleoid region without a membrane. Eukaryotes have a membrane-bound region around their genetic material, called a nucleus. Although we just talked about some differences between DNA and RNA, they do both share a very similar base structure. So this is talking about both types of nucleic acid, DNA and RNA. We're talking about both of them when we're talking about this structure down here. So they both share this general structure in terms of being built by these nucleotide subunits. So each of these is called a nucleotide. And DNA and RNA are built by these nucleotide subunits, repeating over and over and over again. Each nucleotide subunit consists of 3 things. Each one has a base, each one has a sugar, and each one has a phosphate group. And we'll see this over the next few slides as well. Here are the 3 components of a nucleotide again. We have a nitrogen base, a pentose sugar, and a phosphate molecule. All these together make a nucleotide. But now let's see how the structure between DNA and RNA differ in their nucleotides. First off, nitrogen base. So the different bases are adenine, guanine, cytosine, thymine, and uracil. Thymine is only going to be in DNA, and uracil is only going to be in RNA. The pentose sugar, DNA has deoxyribose. RNA has ribose. And then both of them have a phosphate molecule. Let's see what these differences look like on this image. So you see the nitrogenous bases for RNA. We have uracil here, so we know that's RNA. The nitrogenous bases for DNA, we have thymine, so we know this must be a DNA nucleotide. Now take a look at this pentose sugar. It has 1, 2, 3, 4, 5 carbons. That's why it's called a pentose sugar. But let's look at the difference between these sugars. On this 2' carbon for RNA, we have an oxygen. On the 2' carbon for DNA, we have no oxygen. Hence the name deoxyribose. So that's where it gets its name from. Make sure you're able to differentiate between the two and know which one is DNA or RNA based on looking at a structure like you see here. So although DNA and RNA share similarities in their generalized structure, they both have that same structure for a nucleic acid made of those 3 components of a base, a phosphate, and a sugar. And so for all nucleic acids, they have a sugar-phosphate backbone. There is a difference, a fundamental difference, in the specificity of that sugar in their backbone. So DNA's sugar-phosphate backbone has deoxyribose as the sugar, and RNA's sugar-phosphate backbone has ribose as the sugar. All right, now let's put together everything that we've learned over those last few slides. This image is showing a 4-nucleotide long segment of DNA. And really the first question here is DNA. Are you sure it's DNA and not RNA? Well, let's make sure. Let's first look at the nucleotide bases. We have adenine, cytosine, guanine, and thymine. So we know thymine is only in DNA. Okay, so this must be DNA. But now let's check that pentose sugar. Here are the 1, 2, 3, 4 pentose sugars in this image. And let's look at the 2' carbon. Do you see an oxygen off of this 2' carbon? What about this one? Or this one? Or this one? No. So this is deoxyribose. That's the sugar in the sugar-phosphate backbone. So this must be deoxyribonucleic acid. So it is, yes, we've confirmed it is DNA. Another thing to note in this image is this phosphodiester linkage or this phosphodiester bond. This is how nucleotides are put together one after another. So that is from the linkage between the 3' end of one pentose sugar with the phosphate that's coming off of the 5' end of another sugar. So it's connecting 3' to 5'. And that's the last thing I want to bring your attention to on this image because it's going to come in handy later when we talk about DNA replication. Notice that the orientation of this segment of DNA has a 5' end and a 3' end. So just keep that in mind. Now, how are the 2 strands of DNA sticking together? We know it has a sugar-phosphate backbone, but what about the nitrogenous bases? How are those sticking together? So we need to understand the rules of base pairing. Cytosine always binds to guanine. Adenine always binds to thymine. And of course, in the case of RNA, thymine is replaced with uracil. But these always bind with each other on either side of that double helix. And note that thymine and adenine share 2 bonds, and they are hydrogen bonds. And cytosine and guanine share 3 bonds. So that means that cytosine and guanine is actually a stronger base pair than thymine and adenine. Here is that double helix DNA structure that I've been referring to. And so let's just locate some things on this image. First off, it has a sugar phosphate backbone, and it has two sugar phosphate backbones because this is a double-stranded molecule. DNA is a double-stranded molecule assuming the shape of a double helix. And all cellular organisms have this double-stranded DNA. There are actually some viruses that have single-stranded DNA, but we're going to focus on DNA is always double-stranded in cellular organisms. And then let's look at these nucleotide bases that are coming off of this sugar phosphate backbone. Cytosine is always bonded with guanine, and thymine is always bonded with adenine. So that is always the case. These bonds are a specific, predictable order of base pairing. This is what allows DNA to replicate itself because it can look at one side of the strand and look and see there's a guanine and say, yes, there should be a cytosine. And that is really the basis of DNA replication. So enough about DNA for a bit. Get out of here. Let's talk about RNA and solidify what we know. We know RNA stands for ribonucleic acid. We know that the pentose sugar is ribose, not deoxyribose. It's single-stranded instead of double-stranded. Adenine bonds with uracil instead of thymine. No thymine in RNA. But cytosine still bonds with guanine. Now pretend for a second that you can't see this. You don't know it's labeled RNA. How would you know? 1, we have a nitrogenous base is uracil. And we know uracil is only in RNA. And then 2, let's look at this pentose sugar. Let's look at the 2' carbon here. Is there an oxygen present? Yes. So we know that this is ribose. So indeed, this is RNA. RNA has a lot of important jobs. If we didn't have RNA, we would not have gene expression. So all of the instructions that are in our genome or a bacterial genome would never get carried out without the help of RNA. So here are the different types of RNA. We have messenger RNA or mRNA. Make sure you know both of these names and that they're interchangeable. If you only see mRNA, don't be confused. It means the exact same thing. mRNA is a copy of a gene. So it's a copy of a gene from DNA. It's a messenger of that gene, and it carries the code for that gene to make a protein. Transfer RNA or tRNA transfers or carries an amino acid to the ribosome. And at the ribosome, which are protein factories, that amino acid gets added in a chain of other amino acids, and eventually that chain of amino acids becomes a protein because proteins are made of amino acids. And don't get too worried about this part yet. We'll talk more about this on a later slide. And then ribosomal RNA or rRNA. This is the easy one. This is just a component of the ribosome. So the ribosome is not made completely of protein. It also has this ribosomal RNA region. Now let's jump back to DNA. And let's ask the question, what does DNA do? Why do we even care about it at all? So DNA stores our genetic information. It's our genetic blueprint for being a living thing. For us, for bacteria, for all living things. It's all the instructions on how to be whatever organism you are. And DNA has these 2 major things it does, these 2 main roles in the cell. It needs to be able to replicate, so cells can make more of themselves and make copies of themselves and pass on genetic material to dividing cells. And then gene expression. So actually taking those instructions in the genetic material and then making some final product about it. And that consists of 2 steps that we're going to talk about later on, transcription and translation. DNA and RNA together are involved in this flow of genetic information, where genes in DNA can be expressed by turning into an intermediate form called RNA, and then being turned into a protein at the ribosome through these processes called transcription and translation. DNA can be recombined This is going to help bacterial cells evolve, change, overcome things in their environment. Think back to the previous lecture on cell structure when we talked about bacterial conjugation and the sex pillus. This is an example of recombination and bacterial cells sharing genetic information. And finally, we have replication. For microbes, or for any cell, to divide and make more of themselves, they need to be able to replicate the genetic material and give a copy of that genetic material to each daughter cell. So each cell ends up being a fully functioning cell with all of its instructions on how to be a living thing. So during this lecture, we're going to dive deeper into each of these processes, starting with DNA replication. The central dogma of molecular biology. This is an important rule that states that the flow of genetic information only happens in one direction, from DNA to RNA to protein. And we'll revisit this slide whenever we talk about gene expression. But for now, we're going to focus on this part right here, replication. Because bacterial cells need to make more of themselves, they're going to go through a certain process to do that, and that process is binary fission. And if you remember, binary fission looks a lot like mitosis, but it's not mitosis because we're talking about bacterial cells or prokaryotes. Prokaryotes go through binary fission, eukaryotes go through mitosis. But we're going to, either way, we're going to end up with two cells, and so we need to make a copy of that DNA. And how are cells going to accomplish that? That's what we're going to take a closer look at now. But before we can even begin replicating DNA, remember that DNA is a double-stranded molecule. So how are we going to get to these nitrogenous bases that are locked away in here? First, double-stranded DNA has to be separated into two single strands. So this is a double-stranded strand up here. We have 2 strands, and we're going to separate it into these 2 strands down here. This exposes these nitrogenous bases, or it unzips the DNA molecule, and turns each strand into a template for replication. And it's able to act as a template because of those strict rules of complementary base pairing. So adenine always binds with thymine, and cytosine always bonds with guanine. This mode of replication is referred to as semi-conservative because the result is 2 molecules of DNA that each have a strand from the parent strand. So this was the parent strand, and it unwrapped, and now each one has a strand from the original, and each one is creating or synthesizing a new strand. So the original parent strand is semi-conserved in these daughter strands. Now over in the next couple slides, we're going to kind of dive into how this process works at a cellular level. We'll start off by introducing you to some important players and rules involved in DNA synthesis or replication. You can use those terms interchangeably, so DNA synthesis or replication. Now, DNA is unzipped by DNA helicase. Because it's DNA's double-stranded molecule, we have to open it up. DNA helicase is going to do that. Now that those strands are separated, now they can be replicated. DNA is copied or replicated by DNA polymerase. And DNA polymerase has a special rule where it can only move in the 5' to 3' direction. And this directionality, remember, I mentioned this towards the beginning of the lecture, comes from that sugar. So the 5' and 3' direction is related to that sugar in the sugar phosphate backbone. But just remember, DNA polymerase can only move in this direction. It has directionality. And because it only moves in this one direction, that means we're left with a leading strand that is synthesized or replicated continuously, nonstop, and a lagging strand that's synthesized discontinuously, resulting in Okazaki fragments, or these spaces of DNA that were not successfully replicated. Those gaps in the DNA are then filled in by something called DNA ligase. And these strands are because DNA has this directionality where it's 5' on one side, 3' on the other. And then the other strand runs in the opposite direction, 5' to 3' on this side. So this is the orientation of a DNA molecule. And this is we're going to see on the later slides, this is where that leading strand and lagging strand come from. The last thing I want to bring your attention to on this slide, let me grab my highlighter, is this. This ends in ASE, this ends in ASE, this ends in ASE. DNA helicase, DNA polymerase, DNA ligase. These are all enzymes. So there are a lot of important enzymes involved in DNA synthesis. Now let's look at these enzymes at work. So right here we have DNA helicase, and DNA helicase is unzipping this double-stranded DNA molecule, and at the point of the molecule where it's unzipping, this is called the replication fork. So it's unzipping this double-stranded molecule. Now we've opened it up and we have 2 strands. Now we have, each one has a template strand from the parent that is going to act as a template to make something called a complementary strand. So each of these also has a complement. So the complementary strand is the one that's being added on by DNA polymerase. So the complementary strand here, and there's a complementary strand right here. So that's the new strand. And DNA polymerase is the one that is adding on these new nucleotides. So we have these free nucleotides that are being added on to the template strand, forming a complementary strand with the help of DNA polymerase. DNA polymerase has directionality. It only moves in this 5' to 3' direction, so it's only adding nucleotides onto the 3' side. So we have 5' to 3', so it's only moving in the 5' to 3' direction. This DNA polymerase has to move in this direction in order to follow the rule of 5' to 3'. So this DNA polymerase can only copy so much of the molecule as it's being opened up. So as this molecule opens up, it'll sit down and copy, but look, it'll run into what it's already copied and it will fall off. And that's why this is the lagging strand, and it's moving discontinuously. Whereas the leading strand, this DNA polymerase is able to attach to the molecule and just zoom along, and as helicase unzips more and more of the molecule, it will just follow helicase all the way down the molecule. The next slide will give a better view of kind of these Okazaki fragments and how DNA polymerase works with its directionality as well. Now again, this lagging strand is synthesized in these small fragments called Okazaki fragments. So here's an Okazaki fragment. Here's an Okazaki fragment. And DNA polymerase sits on the molecule on this template strand and then adds the nucleotides in that predictable order, because adenine only binds with thymine, cytosine only bonds with guanine. And it moves along, and eventually it will just run into what it already replicated and it will fall off. And DNA polymerase is such a bulky molecule, it will fall off before it's able to replicate everything. So it leaves these gaps, these open bits of DNA that were not replicated. And that's where DNA ligase comes in. So DNA ligase just joins those discontinuous fragments and fills in those gaps. You can also see here again with the directionality. DNA polymerase always moves 5' to 3'. Here's the 5' side. Here would be the 3' side. So we know that this is the continuous strand. DNA polymerase is able to just zoom along here as helicase unwinds the double-stranded molecule. Something to point out here though. Remember that we are talking about bacterial cells primarily, or prokaryotes. So prokaryotes have circular genetic material, so their genomes are circular. That means for prokaryotes, they're going to form something called a replication bubble, and they're going to have to open up a part of their circular genome, and then they can move in both directions simultaneously, left and right, to replicate their genome. Another image showing the directionality of DNA polymerase. I just feel like this is one of those kind of difficult concepts that's difficult to kind of nail down, because you get confused, or you might get confused, maybe not, about which side DNA polymerase is adding nucleotides to. It's always adding nucleotides to the 3' side. So we're talking about, whenever we're saying 5' to 3', we're talking about the complementary strand is 5' to 3'. Always, same thing down here. These complementary fragments, this is the lagging strand because we've formed Okazaki fragments, are again 5' to 3'. So now we know DNA polymerase is involved in adding those nucleotides onto the growing DNA molecule. But how are nucleotides actually being added? If you think about this for a second, is this process anabolic or catabolic? We are building or growing this DNA molecule, so that means it must be anabolic. We are building a bigger molecule from smaller building blocks, so this requires the input of energy. And we're trying to build this bond right here. So it's actually pretty interesting. Each nucleotide comes with its own little packet of energy it uses to be added to that growing DNA molecule in the form of phosphates. So it gets added and it loses 2 phosphate groups through the process of hydrolysis, and it's the breaking of those bonds that it uses as energy to build this bond and build this new DNA molecule. But this should look kind of familiar. We've seen another molecule that had 3 phosphate groups and was used for energy. And that molecule is ATP, or adenosine triphosphate, the energy currency of the cell. And here's the structure of ATP. ATP has ribose. ATP has these 3 phosphate groups. And ATP has adenine. So you should be thinking, are you telling me that ATP is a nucleotide? Absolutely. And we'll see on these next couple slides to kind of explain why that is. But ATP is that same form of adenine that's being added into the growing DNA chain. But instead of losing 1 phosphate group and becoming ADP, or adenine diphosphate, it's losing 2 phosphate groups to be added to a DNA molecule. So whenever it's used as energy currency, it only loses this outermost phosphate. When it's being added to a DNA molecule, it loses both of these phosphate groups. So free nucleotides, adenine, thymine, cytosine, and guanine, provide their own energy to be attached to that growing DNA molecule in the form of those 2 phosphate groups. So it loses 2 phosphate groups as it's coming up to be added to the chain, and this provides the energy to make this bond right here with this oxygen on the 3' carbon of the pentose sugar. Because remember, we still have directionality. We have 5' to 3'. They're always being added to the 3' side of that sugar. And then we're losing 2 phosphates, but we're keeping 1, and that is ultimately going to give us this sugar phosphate backbone of DNA and RNA. So now I hope you're wondering, why is ATP the primary energy currency of the cell? Why not TTP with thymine, CTP with cytosine, GTP with guanine? And for that, I want you to look at the structure of each of these nitrogenous bases. Guanine, we have oxygen present. Cytosine, we have oxygen present. Thymine, we have oxygen present. There's no oxygen present in adenine. So adenine is the only nitrogenous base without oxygen. And this is why ATP became the primary energy currency of the cell, because millions of years ago, whenever organisms were evolving and using energy, oxygen was not as available in the atmosphere as it is today. Oxygen was very scarce. So they evolved to use the nitrogenous base that did not have that limiting factor of oxygen. Pretty fascinating stuff. Now back to this image, the central dogma of molecular biology, because now we're going to talk about gene expression. So I really want you to know the central dogma of molecular biology. I guarantee that this is a question on the exam, because understanding that genetic material can only flow in this one direction in order to make proteins is really important. We need this intermediate mRNA step. So DNA to RNA to protein, always. DNA is transcribed into RNA. RNA is translated into protein. A good way to remember that, if you can't remember which one comes first, transcription or translation, you can just notice that it is alphabetical order. Transcription would come before translation in a dictionary. Transcription is first. So gene expression. We're trying to make proteins from information that's stored in DNA. So DNA has genes that have instructions on how to make the proteins, and now we need to get proteins out of it. And so this whole process is gene expression. And the enzyme that is really important in this process is RNA polymerase. RNA polymerase is what is actually going to be converting or transcribing a gene in that DNA into mRNA. So a lot like DNA polymerase, it does have directionality, and RNA polymerase can only move in the 5' to 3' direction as well. So gene expression involves these two steps, transcription and translation, with the whole goal being to express genes in the DNA. And we accomplish this by converting those DNA instructions into a functional protein with the help of RNA. And so this is kind of the whole journey we're going to be going on through these next few slides. And I want you to know that this image kind of makes it look like this RNA strand is the same size as this DNA strand, but that is not the case. RNA, this would be mRNA, these strands are actually very short because it's only a bit of the DNA genome. It's only coding for a gene within that genome. So it's a very small part of the genome that's coding for a specific protein. So when RNA polymerase turns DNA into mRNA, it's only taking a bit of that genome. Then that mRNA is going to go on its journey to find a ribosome, and at the ribosome, it's going to be translated into a protein. So here, that's the journey we're going on over these next few slides. First up, we have transcription. And for transcription, we are talking about this enzyme right here, RNA polymerase. RNA polymerase is going to transcribe from the language of DNA to the language of RNA. Because RNA is different from DNA, right? DNA has deoxyribose, where RNA has ribose. DNA has thymine, where RNA has uracil. So keep that in mind. We are transcribing from kind of one language of DNA to another into RNA. RNA polymerase is going to sit down on something called a promoter. I'm not going to ask you about this, but it's just the beginning of a gene. And a gene codes for a protein in most cases. So it's going to sit down where this gene starts, and it's going to start copying this length of the genome, the whole length until it reaches something called a terminator, or the end of the gene. And it's going to make this whole length of mRNA. So this is all mRNA. And this mRNA codes for a protein. It's a copy of the gene in this DNA and codes for a protein. After our genetic information has passed from DNA to mRNA, we've transcribed it from DNA to mRNA. Now we're going to translate it into the language of a protein. So we transcribed into the language of mRNA. Now we're going to translate into the language of proteins. And we're going to build up proteins with amino acids. Remember that amino acids are the building blocks of proteins. So this mRNA strand that RNA polymerase made for us by copying a gene in the genome is read in segments called codons. So it's read in these groups of 3 nucleotides called codons. Each codon is composed of 3 nucleotides. Each codon is a code for a specific amino acid. So each codon represents an amino acid, and this is how the mRNA strand is read whenever it goes to the ribosome. It's read in this frame called a reading frame. So each codon is also called a reading frame. And this reading frame scoots along. Oh gosh, I promise I'm trying really hard to write well with this pen. And this reading frame is what's read as the mRNA scoots along the ribosome. And we'll see the transfer RNA brings in the appropriate amino acid that codes for what it's reading on the mRNA. And that's how these amino acids are built together to ultimately build a final functional protein. So here's all the different nucleotides that can come together to code for these proteins. And you'll notice that there are more than 20 spots here, right? And you should be thinking, but there's only 20 amino acids. There are only 20 amino acids, but the code or the codon code that's in that mRNA is actually a degenerate code because you can have more than one codon that codes for a type of amino acid. Like you'll see here that the first position U followed by the second position U followed by the third position A, UUA codes for leucine, and also UUG codes for leucine. And all these other things down here code for leucine as well. So it's a degenerate code because more than one codon can code for an amino acid. But notice here, though, that you do have a first, second, and third position here because codons are composed of 3 nucleotides. Great, so now we have made our mRNA. RNA polymerase has copied a gene in the genome, made it into mRNA, and now we're going to see what happens at the ribosome, where we translate that mRNA to make a protein. And remember that ribosomes are located in the cytoplasm of both prokaryotes and eukaryotes. I really like this image that I just found because it shows very clearly what attributes are shared between prokaryotes and eukaryotes, and you see both of them have ribosomes for protein synthesis. Both are located in the cytoplasm. The difference here, though, is in a prokaryote, since we don't have a nuclear membrane, they only have the nucleoid region, this mRNA didn't have to pass any membranes to get to a ribosome. It was turned into mRNA and it was able to just go boop, straight to a ribosome. But in a eukaryote, the genetic material is encased by a nuclear membrane. It's inside of a nucleus. So that means the mRNA that was made in the nucleus, where the genome is, had to be transported across the nuclear membrane first before it could get access to ribosomes in the cytoplasm to turn into a protein. Now we've got our mRNA to the ribosome to build our protein, but how is that going to happen? How are proteins actually being built? And the thing you really have to ask yourself to go into that question is, what are the building blocks of proteins? And that, of course, is amino acids. Amino acids are the building blocks of proteins. So how are we going to get amino acids to the ribosome to build the protein that has for the instructions that are on that mRNA? And that's where a special kind of RNA comes in called tRNA, very special molecule, tRNA or transfer RNA. And transfer RNA's job is to transfer amino acids to the ribosome, to read the instructions on the mRNA and bring those amino acids to build the protein. And you'll see this kind of artistic rendering of tRNA right here. We kind of think of it as this more simplified version. It is more complex in real life, of course, but on this version you can easily see the 3' end where amino acids would attach to tRNA, and tRNA will carry these amino acids to the ribosome. And at the ribosome, there's a specific amino acid for each type of codon on the mRNA strand. And we'll see this more on the next slide, but I just want to point this out before we move on. tRNA also has an anticodon region right here. It has one to 3 nucleotides in its anticodon region. And we just saw this word recently, codon. Codons are the 3 nucleotides on the mRNA strand that create the reading frame to read the amino acids that are used to build the protein that it codes for. So let's look at the next slide and see this in a bit more detail. Here we're seeing this anticodon region again, and its relationship with the codon on the mRNA. So this mRNA has this codon that's composed of 3 nucleotides that is UCA. This codon is going to bind specifically to a tRNA molecule that has the anticodon AGU, because A binds with U, G binds with C, and A binds with U again. And this tRNA is carrying the amino acid that codes for this codon right here, and it's carrying it to the ribosome, and then this tRNA will transfer this amino acid to the protein to build it, and then it will go off and get another amino acid. But just for this slide, pay attention to this relationship between the anticodon and codon region. Know that anticodon is on the tRNA, codon is on the mRNA. Now all of this is taking place at the ribosome in the cytoplasm of prokaryotes and eukaryotes. And so ribosomes are these protein factories where proteins are being built. And just a couple things to point out. Ribosomes do have a large and a small subunit. And these 2 subunits are actually sitting on the mRNA. Whenever the mRNA strand comes to a ribosome, the large and small subunits will attach onto the mRNA and move along that strand of mRNA to read it and make that protein. And then just make sure you keep in mind that prokaryotes and eukaryotes do have different ribosomes that do the same job. Prokaryotes have a smaller molecular weight ribosome compared to eukaryotes, but just know they both have ribosomes. All living things need to make proteins, but they are slightly different in terms of their molecular weights. So here's the actual process of protein synthesis. We're putting all of this stuff together. So we're thinking ribosomes, we're thinking mRNA, we're thinking tRNA, we're thinking amino acids. All of this stuff has to come together. So to start synthesizing a protein, ribosomes will sit down on the mRNA because it has that top and bottom region. It will sit down on the mRNA and it will attach to a start codon. So we know it's the start of a gene or the start of the protein instructions. And then elongation will happen. The ribosome will actually move along the mRNA. It'll go from codon to codon, reading that codon. And as it moves from codon to codon, the tRNA will bind to each codon with its anticodon region and bring the specific amino acid that the mRNA is coding for. It'll bring the amino acid and then it'll add those amino acids to the growing chain one by one by one. And that growing chain of amino acids eventually becomes a protein because proteins are made of amino acids. Eventually, the ribosome will come to something called a stop codon, telling it that that's the end of the instructions for the protein, that's the end of the gene, and the ribosome will then unbind and release. And then you have a complete protein that can then fold and create a specific 3-dimensional shape and become a functional protein. So here are those steps put in action. We have our mRNA strand. We have our ribosome, which is attaching to this mRNA strand. We have these codon sites on our mRNA. They're made up of 1, 2, 3 nucleotides each. That's a codon. And then we have our transfer RNA. So this is our tRNA right here. And it's doing its job because it's transferring amino acids to the ribosome. MET is methionine, so it's a start amino acid. That's what kind of starts a protein. So this transfer RNA is bringing this amino acid to the ribosome to start off building this protein. So now let's see what happens as tRNA keeps reading along this mRNA strand and bringing more and more amino acids to the ribosome. So we saw on the previous slide tRNA carrying methionine, this start amino acid, to the ribosome, and it binds. And now the ribosome's moving along this mRNA strand, and now we're getting to the next codon. Our next codon is GGU, and that's going to code for glycine. So this tRNA is going to come along. Its anticodon region on the tRNA is going to bind to the codon region of the mRNA, and it's going to transfer, the original tRNA is going to transfer its amino acid to this new tRNA that came in. So now we have two amino acids together. We have glycine and methionine. And now the ribosome is going to continue reading along mRNA. A new tRNA is going to come in. This time we have alanine coming in. And so this growing amino acid chain is going to attach to the new amino acid, and we're going to end up with a methionine, glycine, alanine chain. And this happens along the whole length of the mRNA until the ribosome reaches a stop codon in that mRNA, which tells it that's the end of the protein. And then by the end of it, we'll have a really long chain of amino acids that ultimately make up a protein. And just a note here, so mRNA, even though it's very short compared to the entire DNA genome of an organism, it still can be a pretty long length of mRNA, a long length of instructions for building a protein. And so to be efficient, multiple ribosomes can actually attach to the length of mRNA at once, all moving along in the same direction, a 5' to 3' direction, and all reading the mRNA and growing a protein, and then releasing a complete protein. So it's not just one ribosome per mRNA strand. Multiple ribosomes can be moving along mRNA, reading it, and creating proteins. So here's this whole idea of gene expression and transcription and translation in this one image. And notice here, look at this, we have something called a nuclear membrane. So are we in a prokaryote or a eukaryote? You should be thinking eukaryote because prokaryotes do not have a nuclear membrane. They have a nucleoid region. They would have no membrane around their DNA. But during transcription, so we have our genome, we have RNA polymerase that is converting that DNA into mRNA, these instructions for this gene that creates a protein in the DNA. And then that mRNA that has been transcribed is going to leave the nuclear membrane, since we're in a eukaryote, and head on to the cytoplasm where the ribosomes are located. At the ribosome is going to bind to mRNA on a start codon. A codon is 3 nucleotides long, and each codon codes for an amino acid. And then tRNA is going to bring the amino acids necessary to build the growing protein, because proteins are made from amino acids. Eventually, the ribosome will reach a stop codon and leave the mRNA strand, and then that completed polypeptide chain of amino acids will fold on itself, creating a 3-dimensional shape and ultimately a functional protein. Now think back to towards the beginning of the lecture, I said we were going to talk about DNA replication, gene expression, and genetic recombination. So now we're on to genetic recombination. And genetic recombination can be vertical or horizontal. There's vertical gene transfer and horizontal gene transfer. And vertical gene transfer is talking about between generations of cells. And it's vertical because, I guess you, in this image in particular, you can kind of think of it as vertical, up and down. So we have a parent cell that divided, it replicated to make 2 daughter cells, and that is vertical gene transfer. Genes were transferred from one generation, here's the parent generation, to the second generation down here where we have 2 daughter cells. And that's vertical gene transfer. Horizontal gene transfer, on the other hand, is not transferring genetic material from one generation to another. It's actually happening, you can think of it as the same generation. So it's happening between organisms of the same generation, not transferring by dividing to create a new generation. And the example that we're really going to talk about here is the sharing of plasmids and the sex pilus and bacterial conjugation. Those are examples of horizontal gene transfer because bacterial cells are getting new genetic information from not their parent cell dividing itself, but from somewhere else in the environment or from some other bacterial cell. And genetic recombination and events of horizontal gene transfer, like bacterial conjugation, increase the genetic diversity of prokaryotes. So it increases their genome, it increases the number of genes that they have, and oftentimes genetic diversity really helps with organisms surviving in different types of environments. So this image right here is showing bacterial conjugation because we have a conjugation pilus or a sex pilus, and there is a plasmid here that is going to be shared between these bacterial cells. And remember, a plasmid is a circular small piece of extra genetic material, so it's also made of DNA, and most of the time it's going to exist outside of the chromosome or the main genome of the organism. The plasmid can integrate into the genome, but oftentimes it just exists outside of that genome in the cytoplasm. And whenever an organism has a plasmid present, it is called the F-positive cell. It has the plasmid. An organism without the plasmid is the F-negative cell. You can also call the organism with the plasmid the donor cell. And then the organism it's giving the plasmid to is the recipient cell. Now these two bacterial cells attach to each other through something called a conjugation pilus or a sex pilus. And this donor bacterial cell is going to copy its plasmid because it wants to keep a copy of its plasmid, but it'll make a copy and it'll send a copy over to this new organism through that conjugation pilus. And at the end, they'll both have the plasmid present, so now they're both F-positive cells. And why do bacterial cells want to share plasmids anyway? Oftentimes, plasmids code for different types of resistance genes or resistance factors. So they let bacterial cells be resistant to things like antibiotics that would normally kill it in the environment. So they're able to share this genetic material between different cells. This is showing that same thing that we just talked about, just in a different artistic rendering. And you see that we have the sex pilus again, or the conjugation pilus. In this image right here, it's called the conjugal junction, but you can still see the word conjugation here. And we have two bacterial cells. One has a plasmid present, and so it's the F-positive cell. This one has no plasmid present. It's the F-negative cell until it has received a copy of the plasmid. And I do want to mention here that once a bacterial cell receives the plasmid, it is then able to transfer that plasmid onto daughter cells as well. So this right here, conjugation, is horizontal gene transfer. But once it has the plasmid, it's able to transfer this genetic material onto its two daughter cells once it replicates, and that would be vertical gene transfer. So replication or binary fission is vertical gene transfer, replicating the genetic material. Horizontal gene transfer happens between cells of the same generation, so we're not making new generations anymore, and it's transferring often, we're talking about plasmids or resistance factors to other bacterial cells.